Hello everyone, I am here again to discuss about normal cholesterol metabolism, and in this video we will cover these topics: what is the basic knowledge of cholesterol, how the cholesterol synthesis are regulated, and what is the role of this steroid regulatory or element binding the proteins, plasma transport and regulation of absorption, what are the types of lipoproteins, endogenous synthesis of the cholesterol and low density lipoproteins, and what is the pathway of the low density lipoprotein receptors, and the feedback the control, and we will discuss about in this lecture, and let's dive into the video. Cholesterol, and this is a steroid type of lipid, and this is essential the structural component of animal cell membranes. And cholesterol, this also serves as a precursor for biosynthesis of steroid hormones, vitamin D and bile acids. In vertebrates, liver cells usually produces a large amounts, and it is absent in prokaryotes like the bacteria and the archaea. And cholesterol may be derived from our the diet or from endogenous synthesis, and we will discuss all these things in this lecture. Regulation of cholesterol synthesis. The biosynthesis of the cholesterol is directly regulated by cholesterol levels that are present, and the main regulatory mechanism is the sensing of intracellular cholesterol in endoplasmic reticulum by the protein SREBP. In the presence of the cholesterol normally, and this is the bond to two other the proteins, SCAP and INSG1, and SCAP is abbreviated as these SREBP cleavage activating proteins, and the other is the insulin induced G1 the protein. The proteases S1 site 1 proteases and site 2 the proteases, and they are actually activated by the SCAP, and when cholesterol levels are low in our the body, and then the action is that these proteases are usually involved in the cleavage of SREBP the protein. The other thing that you should know is about HMG function, HMG coenzyme A reductase, and which is a rate limiting enzyme, and overall its action result is to increase the level of the cholesterol synthesis. And so when the cholesterol levels are high in our the body, the rate of degradation of HMG coenzyme reductase are reduced by tenfolds. Here you can see here that in endoplasmic the reticulum, ISNG is the attached to the SCAP and SREBP. When cholesterol levels they falls, and this ISNG1 that dissociates the SREBP, and this SREBP and SCAP the complex, and which allows the complex to migrate to Golgi the apparatus, and here the SREBP is cleaved by these two the proteases S1P and S2P. The cleaved SREBP then moves towards the nucleus and moves into a nucleus, and where they act as a transcriptome the factor and binds to SRE, which stimulates the transcription of many the genes, and among these are low density lipoprotein receptors and HMG coenzyme A reductase. LDL receptor, they captures circulating LDL from the bloodstream, and whereas HMG coenzyme A reductase leads to an increase of endogenous production of cholesterol. And other main things you should know that even when a cholesterol the synthesis can be turned off when the cholesterol levels are high, and the role is that HMG coenzyme reductase have both cytosolic the domain, and which functions responsible for the catalytic the function, and there is the membrane the domain. The membrane domain senses signals for its the degradation. Increasing the concentration of the cholesterol cause a change in the domain's oligomerization state, and which makes it more susceptible to destruction by the proteasomes. Plasma transport and regulation of absorption. And cholesterol and triglycerides, they are insoluble in water, they are non-polar lipid substances, and so they have to be transported in a plasma. And so according to the density of increasing the density of the lipoproteins, and they are the divided into the chylomicron, very low density lipoproteins, intermediate density lipoproteins, low density lipoproteins, and high density lipoproteins. And chylomicrons, different triglycerides, and the cholesterol, they have to be incorporated into chylomicrons in order to travel into the blood. And so they use the gut lymphatics to travel into the blood. And they carry fats from the intestine to the muscles and the tissues, which are in need of fatty acids for energy or fat production. And these chylomicrons are then hydrolyzed by endothelial lipoprotein lipase, and are present in the capillaries of the muscles. And unused the cholesterol remnants, they are more cholesterol rich chylomicrons and aminoacids, because the fats they have the delivered to the muscles and the other tissues, and then they are from the hair, they are taken up by the liver to the stream. And some of the cholesterol enters the metabolic the pole, some is excreted as free cholesterol or as the bile acids into the bile tract. Next, we will discuss about endogenous synthesis of cholesterol and low density lipoproteins. And this the endogenous synthesis begins in liver, and the first step that liver performs is the synthesis of very low density lipoproteins, and which are then undergoes the lipolysis, and they are converted into the intermediate lipoproteins. And in comparison with the very low density lipoproteins, and the content of triglycerides in IDL is reduced, and that of the cholesterol is the enriched in intermediate the lipoproteins. And intermediate the molecules are then consumed in the either to the processes. Most of particles are either directly taken up by the liver through LDL the receptors, and while the others are converted to into the cholesterol rich LDL form for the further the loss of triglycerides and the loss of apolipoprotein E. In a liver cell, IDL is recycled to generate very low density lipoproteins, and again then again this cycle is repeated. The LDL receptor the pathway, and this metabolizes usually about two-thirds of resultant LDL particles, and about 75% of these are located on the liver cells, and that's why liver plays a very important role in lipid metabolism. LDL receptors binds to apolipoproteins B100 and E, and thus is the involved in the transport of the both LDL receptors and intermediate. So the first step in receptor mediated transport of low density lipoproteins involves the binding to the cell surface, and the ligand binds to these receptors, and then which undergoes the internalization inside the so-called the clathrin coated the pairs. And within the cell, these cytotic the vesicles the fuse with the lysosomes, and the LDL molecules is the enzymatically the degraded. Free cholesterol is released into cytoplasm, and the cholesterol is not only used by cell for membrane synthesis, but they also take part in intracellular cholesterol homeostasis by a system of the feedback the control. It suppresses actually cholesterol synthesis by inhibiting activity of HMG coenzyme A reductase. It stimulates the formation of cholesterol esters for storage of excessive cholesterol, and it down-regulates this synthesis of cell surface LDL receptors, and thus preventing excessive accumulation of cholesterol inside cells. And so that is all about the cholesterol. Actually, I was going to make presentation about familial hypercholesterolemia, and for that you have to understand the cholesterol basic the concepts, and that's why I made these presentations. Hope to see you again. Thank you very much.